Part 3. Okay, part 3. Expiration date and nature of the expiration date. Connection between surgical site infections and reprocessing of medical devices, and control of reprocessing processes. Expiration dates. The expiration date of sterile material depends on the selected storage environment, type of storage, and packaging system. Expiration date is not connected with the sterilization method. All sterilization methods must reach the same reduction level. Please never think about that sterilization method makes a storage date longer or shorter. Only packaging system, where it is stored and how it is stored. And the expiration date of sterile medical devices is determined using the point method. Point method was established by Dutch engineer Jack van Asten. It was proceeded or done in 1990s, and he spent 6 years for this. So, checking how long we can store in sterile stocks instruments when they start to be non-sterile. Expiration date means not how long surfaces of the instrument is still sterile, but it means how long barrier system can guarantee that medical device quality is not changing. How long barrier system or packaging material can guarantee. After this, when expiration date is over, it doesn't mean that the instrument inside is non-sterile. It means only that packaging system cannot guarantee and instrument surface starts to stay non-sterile. How fast it is going on, it depends on environment around. Connection between surgical site infections and the reprocessing of medical devices. Surgical site infections, it's one of the most popular hospital-acquired infections, and World Health Organization says it's approximately 0.5 up to 3% of all patients who have surgical manipulations. But according to European Operating Room Nurse Association, approximately 37% of all hospital-acquired infections are caused because surgical site infections. Approximately 37%. And here is a different possible reasons. It can be dirty team, dirty environment around, dirty equipment, dirty instruments, transportation ways after surgery to department, and of course care in the department. Tricky is that it's really difficult to say where is the real reason why it happened, because after manipulation rooms are cleaned, maybe even disinfected, team at least washes hands, and instruments are reprocessed. So we practically have no evidences. What we have is patient itself care in a department after this manipulation, and of course it can be also transportation ways. It's really tricky. It's difficult to say where is the real reason. World Health Organization says, okay, instruments are involved in approximately 0.5 to 1.5% of all surgical site infections. Instruments are involved. But in reality, the opinion is that it's approximately 10%. Roughly 10% of all surgical site infections are directly or indirectly because of surgical instruments. And of course it can happen. It can happen because of incidents during reprocessing, and it can happen because reprocessing is done by human. Incorrect opening in the user site, and it also can happen. It's very rarely, but it can happen. Not correct opening of packages and not correct presentation. Also important is improper or wrong storage in a user site. And what is very important, it's time gap between manipulation and reprocessing. As longer is this time gap, as higher are risks for the next patient. Because microorganisms are surviving and they are multiplying. Yeah, I think you know. And as longer is this time gap, as higher are risks for the next patient, because a reduction is only amount of times. And it can happen really with a perfect process, with a perfect reprocessing process. It can happen that if starting amount is so huge that after this process the rest is quite enough for new infection. It can happen. Control of reprocessing processes. Yeah, what you have to remember that there are no non-destructive methods for the control of the result of the reprocessing processes of medical devices. No. And why? When medical devices are reprocessed, before the last step, before sterilization process, they are packed and packages are closed. So it means technicians who are making decisions, is it allowed to send to you, to the doctors, or no? They have no possibilities to measure. What they can do, just check indicators or indications about the processes. Okay, we have one possibility. It's, we can send them to laboratory. Laboratory will take the measurement, but they will open those packages. Instruments now are not sterile, and we have to proceed one more time. And you will never get your instruments. Even if laboratory says, yes, surfaces were sterile, but we have to reprocess one more time. Yes, we are using different types of indications. Different types. It's different protein tests, chemical indicators, biological indicators, machine printouts, connections to the computers. Protein residue tests, it's direct and objective tests, but we are using them only while we are checking washing disinfection machines. Are they working correctly or not? Because it's direct test, directly from the surface of instrument, but only for washing and only several instruments. Chemical washing tests, indirect tests, because they're showing what's happening with them. and nothing about the instruments which are in the same cycle. Chemical thermal disinfection control tests, also indirect tests, showing was the required temperature reached, was this temperature the required time, but it's indirect. They are showing what was happening in a machine. Those indications are not showing what's happened with the surface of instruments, what happened with the microorganisms on those surfaces. Printouts of the machine also shows what is happening in the machine, and the same is also with the computer printouts. Machine printouts are showing this with the numbers, computer printouts with the graphs. But it's the same. It's only showing what machine thinks about this process, but not in reality what is happening with the microbes or microorganisms on the surface. Chemical sterilization process control tests, also indirect tests, shows only were parameters reached or not, temperature, time, and active agent. Was it or not? And no more. But they are used as daily tests. Biological indicators, also indirect. Okay, here we use bacteriospores for this, but they shows only, yes, in this indicator we destroyed bacteriospores, but what happened with the instrument surface we don't know. Of course, the same is with the printouts and computer connections. And that is why sterility is only a probability, because all those indications except laboratory tests and protein residue tests, they are indirect. All those indications shows how machine worked, but no one indication shows what happened with the instrument surface, what happened with the microbe on the surface. That's what you have to remember. All those indications are indirect, and sterility is only a probability. Yeah, as I said, it's only a probability because all those indications are indirect. They are not showing a real situation with the instrument surface. They are not showing a real situation with the microbes on the surface. So, and now references, you can just see information about this. Yeah, and thank you for your attention. Thank you if you listen to this, and I hope you will remember about this, that sterility is only a probability. You will remember that regulations are for all of us. Yeah, we have to follow them. So, good luck.